Hello students. In our previous video, we learnt about control and coordination in animals. In animals, coordination is achieved by two methods. First, chemical coordination through hormones of endocrine system. And second, electrical coordination through nervous system. As you know, plants lack nervous system. So, coordination in plants happen only through chemical coordination, that is through hormones. The plants also synthesize hormones like animals, but their hormones are known as phytohormones. Like animal hormones, the plant hormones control the growth and development of the plants. Plant hormones can be classified into 5 categories, namely auxin, cytokinins, gibberellin, abscisic acid, and ethylene. Auxins, gibberellin, and cytokinins are growth promoters, and abscisic acid and ethylene are growth inhibitors. But students, note that ethylene is regarded as a multifunctional phytohormone that regulates both growth and senescence. It promotes or inhibits growth and also controls senescence processes depending on factors like its concentration, timing of application, and the plant species. In most plant tissues, ethylene slows down or reduces growth due to decrease in cell division in both roots and shoots. Let us explore each hormone one by one. Auxins were the first plant hormones discovered. Auxin hormone is produced in the tips of roots and stem that promotes cell elongation. Stems and roots respond differently to auxins. Cells in stem grow more while less in roots. We are aware of phototropism, that is, directional movements in plants. For example, the stem of plants bend towards the light source. But have you ever wondered how the stem bends itself, especially when they do not have eyes? The light hits on one side of stem more, and this breaks down the auxin more on this side, while auxin on the dark side do not get broken down and get accumulated on the darker side of the stem, as breaking of auxin is less on this side. So its concentration is less on side where light is falling, while its concentration is more on side where it is dark. And as we know, auxin is growth promoter. Cells of dark side grow fast and more. As you know that the auxins are growth hormones of the plants, this results in the slower growth on the bright side and faster growth on dark side, causing the stem and the leaves to bend towards the side of light. You will recall that this is the process of phototropism, where photo means light and tropism means directional movement. Auxins are classified into two types, namely natural auxins and synthetic auxins. Auxins produced by plants are called natural auxins. For example, indole acetic acid, that is IAA. While auxin produced artificially are called as synthetic auxins. For example, dichlorophenoxyacetic acid. Now let's see the function of auxin. The most significant role of auxin is cell elongation or cell growth. They promote cell division. Auxin also promote apical dominance. Students, the auxin produced by the apical buds suppress growth of lateral buds. This is called apical dominance, which in turn make plant grow vertically in the height than horizontal branching. This phenomenon of apical dominance is used by gardeners while giving attractive shapes to flowering plants. Can you guess how gardener is able to maintain the bushy appearance of lawns? They cut all the apical buds, that is, buds present at the top or apex of the branch. Auxin concentration is very high in the apical bud region. When the apical bud is cut off, auxins that are concentrated in this region are also removed, and the little birds are no longer inhabited for growth. So the plants attain a bushy appearance instead of height. Abscission is the process by which plant drops its fruit, flowers or leaves. The auxin prevents abscission. Auxins reduce root formation to concentration and inhibit root formation at their higher concentration. Auxins are used as external application to produce seedless fruits, for example, watermelons, grapes, lime, etc. Let's move to another important plant hormone called cytokinins. Cytokinins were first discovered in the milky endosperm of coconut. Cytokinins are a group of growth hormones whose primary effects is to stimulate cell division. Cytokinins are found in plant roots, young leaves and seeds. They are also found in the meristematic regions of the stem. Students, do you know meristematic regions of plants are regions that have cells which can divide throughout its life? Like auxins, cytokinins are also of two types, natural and artificial. Zeatin is an example of natural cytokinin, while kinetin is synthetic cytokinin. Cytokinins together with auxins regulates root and shoot development. If the shoot of cytokinin is more, then the shoot development takes place, and if the proportion of auxin is more, then root development takes place. Cytokinins can help plants to overcome apical dominance and promote lateral bud formation. As you will recall that auxins cause apical dominance. Cytokinins delays the process of aging in plants. This process of aging is known as senescence. They also promote chloroplast development, which helps plant in photosynthesis. Now let's see about gibberellins. Gibberellins are the most evidently found plant hormones. Japanese plant pathologist Kurosawa was trying to find the causes and effects of the fungal infection in the rice plant. He observed that infected rice plants were abnormally tall. It was later discovered that this abnormal increase in height was due to a hormone called gibberellin, produced by the fungus Gibberellia fusicori. So students, you must have understood how it got its name gibberellin. Since gibberellins are acidic compounds, they are also called gibberellic acid, abbreviated as capital GA. Now look at these two cabbage plants. Cabbage plants are supposed to be short and bushy, aren't they? Then why is this cabbage plant so tall? It's because this cabbage plant was injected with gibberellins. So now can you guess the function of gibberellins? Yes, one of its important functions is elongation. Gibberellins cause stem elongation in plants by promoting internodal elongation. Gibberellins break seed dormancy and induces germination. Like auxins, gibberellins also are helpful in development of seedless fruits. Development parthenocarpy is called parthenography. Observe these plants on your screen. Can you name them? Yes, they are cabbage, radish, spinach and wheat. What is common to all these plants? These plants are long day plants. That is, they require longer days and shorter nights for flowering. Do you know, if we apply gibberellins to these long day plants, the flowers bloom even when the days are shorter. But note that gibberellins cannot induce flowering in short day plants. So students, we have seen some plant hormones like auxin, cytokinins and gibberellins as growth promoters. There are other hormones which are growth inhibitors like ethylene and abscisic acid. We will study them in our next video.